Good morning and welcome to Microbes Without Borders. We have a very special guest this morning, and it's Dr. Jamie Foster. And we will talk to her about all the cool stuff she does, because she teaches astrobiology and she's sent squid into space, and she's done really fun stuff. So good morning, Dr. Foster, and thanks for being with us today.
Thank you, Dr. Oli. It's really a pleasure to be here and be part of this course.
Great. So the one thing what I know about you, again, we've been colleagues for a while, but you work at Kennedy Space Center, so we don't see each other very much. You teach a course called astrobiology, and I have students come to my office and they say, what is astrobiology? What is she teaching in this class? So would you mind sharing this with us?
It's not astrology. So if you want to know about what a Virgo is and a Sagittarius, then you're out of luck. But what astrobiology really is all about is about three major components. Understanding where did we come from? How did this planet come to be? How did the universe come to be? The second part is what are the limits of life now? So how did life evolve? Where can life live? And is there life beyond our biosphere? How do we go about looking for life that we don't necessarily know all the parameters of how hot can you go, how cold can you go, and so forth. And the third component of astrobiology is really about the future. Where are we going as a species? Where are we when someday our sun is going to turn into a gas giant and we are going to have to move? And hopefully our species will be around in the 5 billion or a couple billion years that it's going to take for this to happen. So where do we go, and what technology do we bring with us, and how do we get prepared for that? So understanding how life evolved, where did we come from, where is life beyond Earth, and what are the extremes of life, and also where are we going as a species. Those are kind of the three components that fall under the umbrella of astrobiology.
That's really cool. That's very, very interesting things. We talked a little bit about tardigrades and how they're extremophils and how they can live in space. And we can talk about them a little bit later, but how did you actually get interested in astrobiology? It's not sort of like everybody talks about it and you sort of pick it up in the grocery store, but how did you get excited about astrobiology?
I think it started with Carl Sagan's Cosmos. I was in 5th grade when that came out in the early 80s, and I was mesmerized by that television show. What is the possibility of where life could be, and all the parameters of the solar system and how it formed. And my parents would buy me books about the universe and solar system, and I just got hooked immediately. So I always had this idea that I wanted to maybe be an astronaut, and kind of I wanted to be at NASA somehow in some way. And even though now I'm a professor at the University of Florida, I still work with NASA and I have that opportunity. So it really started out with a young person's curiosity about the universe. And then I started taking classes, and I got really lucky in that I took this class called Environmental Evolution with Dr. Lynn Margulis, who by coincidence is the ex-wife of Carl Sagan. So it's very cosmic how I got interested in all this field. But Lynn Margulis took me on an adventure through all of how life came to be, and is there life on other worlds, and how do we think about the habitability of Mars, and it was all very exciting, and I just got hooked and slowly worked my way and finally got a degree, not necessarily in astrobiology, but in science. And so you can pretty much, once you get your science degree, you can go in so many different directions like I did. And I was able to kind of get a foothold in this field of astrobiology, and it's just been fun ever since to work on this subject.
That's a great story. I know about Lynn Margulis. She was one of the people who was in textbooks. She did the endosymbiont theory, and a lot of people don't know about that, but it's really something really amazing. You want to briefly explain the theory?
The theory is the idea that our cells had the origin of the cell that makes up eukaryotes in humans and animals and plants and so forth, really originated as a symbiosis, as this larger cell that through time phagocytosed or took in smaller microbes, and things like mitochondria for animals and chloroplasts for plants. And over time, instead of just eating the smaller microbe that they took in, there became a coevolution, and eventually this cell became a living organism that retained its symbionts, like the mitochondria and so forth. So This was a very pioneering idea, and people thought she was crazy. She had mostly microscopic evidence. She didn't DNA at that time was more of a was not quite. We didn't have the molecular biology that we have today. So she was very persistent, and she wrote a paper, and it was rejected about a dozen times, but she was persistent. She was very sure that she was right. It was finally published. And since then, we've found all sorts of evidence to support her, from molecular evidence like mitochondria. They have their own little genomes and they replicate just like bacteria. So we have a lot of evidence to support her idea. So she was a pioneer that inspired me, not just in her science, but also just as a great mentor to keep going and help develop my own abilities as a scientist.
That's really a great story, and having a good mentor makes all the difference.
It sure does.
Now you've got your own lab, and you're a mentor to other students, which is very exciting. And you do some really cool research. And one of the research you've done fairly recently, and I've read about, is you're sending squid into space. Why would you send squid into space on a space mission?
Now, it's not just Noah's Ark. It's not that we started with aardvarks and we finally got down to squid. It's not that. It's the reason we're doing this Squid in Space project is to really get an understanding of how microbes affect animals in the microgravity environment. So there's two things that you might want to be aware of with regards to this project. One is that when microbes go into space, they change their physiology. They start to grow faster. They can become more virulent, meaning they can cause more diseases. And on the other flip side, animals also change their physiology, and humans can have their immune systems become weakened. So you kind of have this double whammy here. You've got your immune system becoming compromised, and you have bacteria becoming more virulent. So a lot of people focused on pathogens. How are pathogens changing their virulence, and is this something that we need to be really concerned about as we go and explore space and hopefully get to Mars and beyond? And so what we're trying to do is not just look at pathogens, but it gets back to this idea of symbiosis. We are 90% of our bodies are microbial cells, and these are important to our health and to the normal development of our systems. So we need good bacteria. So what my project is, don't just focus on the pathogens, but also focus on the mutualistic bacteria, the good guys, and ask, are they also undergoing all these physiological changes? Do astronauts have to be worried about their normal, healthy microbes? And since astronauts don't like to be dissected too much, we found that out. So we have to kind of use a model system that can be replicated in many times, has a fast developmental rate, and so forth. So the squid, so we're not so interested necessarily in the squid, other than it's a really easy model, it's very manipulable in the lab, and it's really amenable to rapid experiments and turnover. So that's why we kind of use the squid we've sent that into space to look at the effects. And we're finding that just like humans, the squid undergo a lot of physiological changes, some good, some bad. And we're now really teasing apart the effects of the microgravity, of the bacteria in a microgravity or space environment. And right now we've been doing mostly ground experiments, but we're getting ready to refly them again. And the pilot flight experiment that we flew on the last two space shuttles is called STS-134 and STS-135. That really proved that we can keep the squid alive, that they can colonize. So it was really a pilot study. But a lot of the work gets done on the ground because of the cost of flying up to the space station and up on SpaceX and so forth. So we do most of the groundwork and get ready and get all the parameters set. And hopefully in about a year or two, we'll be flying back up onto the International Space Station to repeat these experiments and really get a handle on what the microbes are doing to animal cells, how they interact, how do they talk to each other, and how is health compromised in any way, and what is this balance between health and disease, not just in the space environment, and that informs us on the Earth too. A lot of what we do in space has what I call terrestrial benefits. So we may do an experiment, and with the lack of gravity, all of a sudden we find out new ways that the bacteria are talking to the animal cells. So we can then kind of bring that information back and learn more about how our own bodies interact with our bacteria here on Earth, not just in the space environment. So we're trying to help astronaut health, by trying to understand what is health in the space environment, and is there something that could go awry, and how could we mitigate those changes? So it's astronaut health, and it's also learning about how these things talk to each other. How do bacteria talk to animal cells, so that we can bring that information back. So it's got this twofold kind of idea to the project. And so, like I said, we're not so interested in the squid per se, but they're really cute, they're easy to work with, and they make a great model for understanding our own bodies.
So the symbiont in the squid, is that mainly the Vibrio that causes the bioluminescence, what you're looking at? Because I don't think our students know much about that either.
Yes,
it's a really cool story.
Yes, this is a symbiosis that has been around for the past 25 years that people have started to looking at it. And it's it's called the bobtail squid. It's found in Hawaii. And I'm forced to go to Hawaii to, collect these animals. That's the hard part about science. You have to travel and go to see all these new places, which is really the perk of the job. So the squid live in the shallow sand flats of Hawaii, and they're colonized, as you said, by a luminescent bacterium, Vibrio fischeri. And Vibrio fischeri live in a special organ that's found within the squid. And at night, the bacteria grow to a large population, and they undergo a process called quorum sensing. And that, when you have a large group of bacteria, that triggers this bioluminescence event. And so the squid has the ability to glow in the dark. And the squid kind of uses it, for the Star Trek fans out there, they use it kind of like a Klingon cloaking device. They shine the light downward, and any predator looking up just sees background moonlight. They just don't see, the animal doesn't cast a shadow. So it hides the animal. So that's the benefit to the squid. Now the bacteria get all these nutrients, they get amino acids, they get a place to live. So it's a win-win for this symbiosis. And my PhD advisor, Margaret McFall-Nike, and her partner, Ned Ruby, who works more on the bacteria side, they really pioneered this system. And it's just taken off in our ability to, because it's one bacterium colonizing one animal, you have this, this monospecific association where you can separate the partners, put the partners together. So it really makes a nice model to look at how beneficial bacteria, how mutualistic bacteria affect the normal development of animal tissues. So that's kind of why we chose this system, and I did my PhD on that system. And now here's a story for you. So when I was a graduate student, I went into my PhD's advisor and I said, Margaret, I think that this system would be great for spaceflight. She's like, you want to launch squid into space? And I said, yes, I think they would be great. She's like, good luck with it. It took me about a decade, but I finally did it. So it's a great idea. So if you have a great idea as a student, hang on to those ideas, because sometimes the time isn't right at the start, but eventually over time you can, maybe make that happen, that dream or that idea to happen. So, and the squid for me has been a fantastic platform to really get at these ideas of symbiosis and how microbes interact with animal tissues and how they affect our normal healthy development. And I think it's been, even though it's a simple symbiosis, I think we can learn so much about how these organisms interact.
That's absolutely fascinating. So, but you haven't gone to space yourself, right?
No, I tried. I applied for the astronaut corps for a couple times around and made it past the first round, but never got an interview. So it might have to save my pennies, and when commercial flight becomes available, I'll have to do that, maybe that route. But there is an astronaut call every few years. So for those people out there who have science background, they're taking more and more mission specialists, and more science is getting done on the space station than ever before. So it's a good time to keep those ideas and those dreams alive.
That's excellent. Another organism that's going out into space, or has gone out into space, are the tardigrades, or the water bears, or moss piglets. And they're really amazing organisms. They can survive outer space. They scramble their DNA and put it back together. Can you briefly tell us about these amazing organisms?
Yeah, the experiments is basically to fly tardigrades, let them expose them to the full-on outside space environment where there's a vacuum, ultimate desiccation. You're getting exposed to UV radiation. So these little animals, they're animals, by the way, very small animals, but they are highly resistant and can survive these space environments. So what are the mechanisms? I don't think we are quite there yet with understanding how they do it. But as you said, they have a lot of DNA repair mechanisms. They're not they're still getting damaged, but they're able to repair their DNA in such a fast and reproducible way. So it's great to have an animal. I mean, most of the time these experiments are done with bacteria and so forth, so it's amazing to see this in an animal.
It's still a lot to learn, so anybody who's interested in astrobiology.
Oh, there's plenty of projects, and you don't have to be a biologist. You can be a chemist, you can be a physicist. It's a really interdisciplinary subject, and I'm working with geologists, geochemists all the time, because we really need to come from different perspectives to get a handle on all the different facets of what makes a world habitable for life, and how is life going to be able to survive the space environment and so forth. So it really takes a wide range of disciplines to come into the field of astrobiology.
Yeah, it's a really, really great job. And you mentioned also that you're traveling a lot for your job. And
Yes.
again, we call this class Microbes Without Borders for a reason, because microbes are everywhere and we can study them everywhere. Another project you're working on is stromatolites. And what is that exactly?
A stromatolite is the oldest life form that we know about on the planet. Stromatolites are bacterial biofilms. Biofilms are basically communities of bacteria that live together. And these stromatolites have the ability to take carbon dioxide from their environment and sequester that and turn that into calcium carbonate. So they're actually making rock. And that rock gets left behind, and they make these little discrete layers. The bacteria kind of grow up, they make a layer of calcium carbonate, they grow up, because they're mostly photosynthetic. So they're drawn to the sunlight. So they leave behind this record of their existence and their metabolic activity. So we have fossils of these stromatolites that go back to about 3.8 billion years. Stromatolites were the dominant ecosystem for 80% of Earth's history. So we're very interested in, again, astrobiology is about where did we come from? How did life come to be? So we use modern day stromatolites as analogs to the past to help us understand these processes. And the two best well-known stromatolite systems are, one is in the Bahamas, which is very close to here, and the other is in Shark Bay, Western Australia. And those two sites have the most, especially in Shark Bay, it's the largest. There are millions of stromatolites there. And it's a World Heritage Site, it's a protected site. And it's just an amazing ecosystem to really get at what are the processes. Again, how are bacteria talking to each other? How are they interacting to form and sequester this calcium carbonate? So not only can they help us with issues about the past, how did we come to be, but they might be very useful. They've dealt with all sorts of climate change. They've been around through high CO2 and low CO2. So how do they adapt to these environments, to this changing environment? So maybe not only can they help us understand the past, but they can also help us understand the present of how do organisms, how do microbes coordinate their activities to respond to climate change, to sequester carbon dioxide as calcium carbonate. So there's a lot of things we can learn about it. And as I said, the two best study sites are the Bahamas and the Australia. I spent too many winters in Boston growing up, so I think I want to stick to warm places,
So do you think they could be used in the future to reduce carbon dioxide, which is a current problem with our climate issues?
Yes, so we could theoretically use them in an applied way. I think that biological carbon sequestration is obviously a major component of the carbon cycle, and we rely extensively on microbes sequestering in the deep sea and so forth. So they are definitely a natural part of the carbon cycle that we could potentially amplify or use. We could even use them on spacecraft to help reduce. Right now we rely on a chemical means to reduce carbon dioxide. So there could be ways to use them in advanced life support on space station as well as here on Earth. They may not necessarily sequester as much as artificial means, like that you can pump the CO2 directly into the soil and so forth. But they do have all this potential, and we're just really scratching the surface of how are they working together, because it's very different microbes that come together to form the environment that will allow this precipitation, this sequestering to happen. So again, we have a lot of questions, not necessarily all the answers, but stromatolites can really serve as a really useful analog to understand modern issues that we have to deal with, like climate change, as well as the past.
That's really exciting stuff. Well, so what gets you up every morning when you come to work? Why do you get excited about? What's the most exciting thing when you come to work every day?
I really love all these projects, and I really love the fact that what makes me a scientist is I don't want to punch a clock, right? We joke that it's you do have to work a lot. Maybe you have to work 70, 80 hours a week, but you can work any 80 hours a week you want. But I really, you work these hours because you love it and you're passionate about it, and I can work on anything I want as long as I can get that funding and I can get an excited student to work on that. I can decide to work on whatever I want, whatever issue excites me. If I want to work on Mars and how do organisms survive the Martian environment, I can do that. It's really, I'm only limited by my imagination and my ability to get funding, which is an issue, but it's really an exciting field, and I have such freedom that you just don't get in industry or work in a more traditional work environment. So this is why I just get passionate about these ideas, and hopefully to help improve the base of knowledge and really help science move forward.
That's an amazing career. That's very, very cool stuff. I've got one more question for you.
Okay.
What's your favorite extremophile?
My favorite extremophile, well, as a community, I have to say stromatolites. I mean, because it's like picking your favorite child, right? You just can't do it. Stromatolites have cyanobacteria and sulfate reducers, and there's all these amazing taxa, hundreds and hundreds of taxa. So I'd have to say, as a community, I have to pick stromatolites. And the main driver is cyanobacteria. I'm just really excited about cyanobacteria. Every breath of oxygen you take, it's mainly because of cyanobacteria out there pumping that oxygen out into the environment. So I have to be grateful to cyanobacteria, because we would not exist if it wasn't for them and all the oxygen they produce.
I thought it was plants who produce all the oxygen.
They do produce a lot, but bacteria produce more. If you just take, and if we ever lost all the oxygen, if we ever lost photosynthesis, the sun went dark and we just lost photosynthesis, we'd be in trouble. We'd lose all the oxygen in our atmosphere in less than 1 million years. So we really need those photosynthetic organisms. But bacteria do most of the work.
That's pretty coal. That
goes with I think all the life cycles and carbon cycles and nutrient cycles. Is microbes are doing most of the work.
That is true. We'll talk about fungi in our next interview, and so there's a lot to learn for us, and a lot of things we still haven't understood, and we need more scientists in the future to make a difference and keep us going.
And reach out, to, if you have an interest in this field, talk to your teachers, because that's how I got started. Is I just kind of went up to Lynn Margulis from my class and said, I'm really excited by this, and do you know of how I can become more involved? And she just kind of was like, come with me. And she introduced me to Margaret McFall-Nye, and I ended up working on the squid, not in space, but I started working on the squid symbiosis. So you really want to reach out. If you have a curiosity, please talk to your instructors, because they know so much, they have a lot of experience, and they can help guide you onto a path. And then it's up to you, but they can help you get started.
Yeah, that's true. You have any other advice for our students in Microbes Without Borders?
Just maintain a healthy curiosity. Go online, read all these stories, and microbes are just amazing, amazing organisms. And there's really no shortage of the potential that they can work on. And if you're interested, again, I would just talk to your teachers and become involved. Business leaders, police officers, firemen, if you all just keep maintaining your scientific curiosity, I mean, it's going to help everyone in the end.
That's awesome. Well, thank you very much for joining us this morning.
I had a great time.
Really awesome to hear about your story.
Thank you very much for having me.
And if you want to learn more about it, Dr. Foster teaches astrobiology, and he's always there, an email away. We'll keep all the information posted.
And I have a lot of my ideas on my work posted on my website, jamiefosterscience.com.
Great. Thank you so much.